Hello everyone, I am Sunita Manoj. I am a faculty in biology. So today we are going to discuss the topic cloning vectors and transformation of our DNA. Okay, end of this topic we will be solving some of the important multiple choice questions also. Fine, so we'll move into the topic now. What is cloning vectors? Okay, so cloning vector, which is one of the genetic engineering tool. Okay, you already know genetic engineering tools consist of restriction enzymes and cloning vectors. Okay, so here in this particular topic we are going to discuss various type of cloning vectors as well as the transformation methods, right? So what is mean by cloning? Cloning means something you are propagating. So here what you are going to propagate? So here you are going to propagate your foreign DNA or the gene of interest. You wanted to multiplicate that particular copy of the gene. Understand? And for that you have to insert into a vector. So that vector will be helping you to carry into the host cell. So wherever you wanted to multiply this particular gene, so that gene will be carried by vector. So that's a function of a vector. So how we can define a cloning vector? So a cloning vector is something which is a self-replicating DNA molecule. Okay, a cloning vector is a self-replicating DNA molecule and which is small in size. Understand? So into this, whichever the foreign DNA you wanted to multiplicate, you are going to insert it. Fine. So we'll see which are the commonly used vectors in genetic engineering. Okay. So here in genetic engineering field we have multiple vectors. Okay. So based upon the size of the foreign DNA you wanted to clone, the characteristic feature or the choice of your vector will be differing. So why? Okay, so you wanted to travel from one distance to some other distance. Okay, so according to the distance, according to your comfort level, you will be choosing the vehicle, right? So whether you wanted to take a 2 carry or 3 carry or multiple carry. Right, so based upon your need you are choosing the vehicle to carry yourself. Here also the same, according to the size of the gene, size of the foreign DNA, you are supposed to choose your vector. So for example you can able to see here plasmids, right? Plasmids is a cloning vector. So you can able to see here something I have written 20 kb. This is the carrying capacity. So whichever the DNA or the gene is having the weight of 20 kb, plasmids can able to carry. Alright, so plasmids is a particular type of vector can able to carry 20 kb size of a foreign DNA. Fine. So the next one, bacteriophages. Bacteriophages can able to carry the size of 25 kb. Understand? So like I told you, you are choosing either 2-wheeler or 3-wheeler or multiple according to the size of the people. Right? So you can able to see the cosmid which is a hybrid vector. So which is a phage plus plasmid. Okay, phage plus plasmid. Phage means what? Bacteriophage. Phage plus plasmid means 25 plus 20. Right? How much you will get? Of course it's 45. So the cosmid's capacity is they can able to carry the 45 kb size. Right? And you can able to see something BAC and YAC. These are all the commonly used cloning vectors in the field of genetic engineering. Why? Can you able to see the number here? 300 kb and 1000 kb. Because of they can able to carry a high amount. Okay, their capacity of carrying the foreign DNA, the size of the DNA they can able to carry is quite high, that is 1000 kb and 300 kb, which is an important question too. Understand? Okay, so I told you are choosing the vehicle as per your need. Okay, so nowadays whatever you are purchasing, you will be seeing the features of that, right? So if you are going to purchase a mobile, right, first you will be seeing what are the features it is having, how much is the how much data it can carry, right? What is the quality of the camera, how much charge it will carry, right? Like that you are going to choose a vector. So there also we have to think about the features of a cloning vector, right? What are all the features your vector should carry? Okay, simply you cannot take any other vector for as per your wish. It should qualify for your work. Okay, the first feature is origin of replication. The vector should multiplicate. Then only you will be getting the foreign DNA whichever you inserted, right? So multiplicating means definitely it should replicate and make multiple copies of your DNA. So you can able to see this picture here. Okay, can able to see the picture here. So here this is a vector which is having a replication origin. Understand? So your vector should carry one gene or else consist of one gene, that is a origin of replication. So origin means origin of replication. So whichever the vector which is having this particular origin, so there the replication begins. You can able to see this picture here. Okay, so this gene will be making a bubble like structure. Okay, bubble like structure, and through that bubble it will be multiplicating or bifurcating into two, replication happening, and you will be getting two copies of your foreign DNA along with the vector. Understand? So origin is a must feature for your cloning vector. So origin or the origin of replication, so that is a sequence of gene where the replication starts. There the vector is bifurcating and giving the multiple copies. Fine. And another important feature of origin. So whichever the vector is having single origin. Understand? If they have only single origin like this, okay, only single origin is there. So what they will be giving? They will be giving less number of copies or low copy number of the DNA you have inserted. Understand? Suppose if the vector is having multiple sites instead of one origin which is having multiple origin, what is going to happen? They will be producing high copy number per cell. Understand? So suppose here only one is there, which is single number, so they will be giving block copies. Okay, so here also one origin is there, here also one origin is there, here also it's multiple origin site will be giving multiple copy of the DNA you have inserted. It will be multiplicating very fast, high copy number per cell it will produce. Understand? So here I have given some example, PBR322. Understand? So they will be giving 25 to PBR322, which is an exclusive cloning vector used in genetic engineering experiment. Okay, so they will be giving 25 to 50 copies of per cell, to 25 to 50 copies per cell. So they will be having those many origin of replication. PBR322 will be giving 25 to 50 copies per cell. So the next one, PUC18. Understand? So if you are using the vector PUC18, you will be getting 500 to 700 copies per cell. So that is the reason I am saying single ORI will be giving low copy number, multiple ORI will be giving high copy number per cell. Understand? So these are the two examples. PBR322 will be giving 25 to 50. PUC18 will give 500 to 700 copies per cell. So this is the most important first feature of a vector. So we'll move into the next one. What is the next feature we'll see? Selectable marker. You know marker pen. Why we are using the marker pen? To highlight something, right? To highlight something important. So there you need something colorful. So you will be using a marker pen. So in case of vector, you cannot put any color. They will not give any color. So other than that, what they have to be a selectable marker. So what is making them to make it selecting from a group? Understand? So you can able to see here it's the PBR 322, which is an exclusive vector I told. Right? So their selectable markers are antibiotic resistant gene. Can you able to see here? Ampicillin and tetracycline is the most important antibiotic. Okay, so this particular vector is carrying antibiotic resistant gene AMPR and TET-R. So these two genes we can use it as the selectable marker. Understand? Okay, so first one is your vector should have an origin of replication. The ori site should be there. The second feature is your vector should have selectable marker. What can be kept as a selectable marker? The antibiotic resistant gene can be the selectable marker. Fine. Now we'll see what is the third one. Cloning sites. Of course, you wanted to clone into your vector, right? So definitely your vector should show the space where you can keep your gene. So you can able to see here the scissors. Okay, so here we are going to cut it and insert the DNA which we wanted to multiplicate. Right? So definitely your vector should have cloning site. Understand? So probably in the experiment, that is in the genetic engineering studies, we need very few recognition or very few sites only. We do not want multiple cloning site. Understand why we don't want multiple cloning sites? So multiple cloning sites means, so while cloning, what you will add? You will be adding the restriction enzymes to cut particular portions. Right? If you have multiple site, what will happen? Your double-stranded or double-stranded circular plasmid will be breaking into so many pieces. Understand? Breaking into so many pieces. Okay, so in this vector where we are going to ligate or clone our foreign DNA. So of course we can able to clone our foreign DNA in the antibiotic resistant gene. So in case of PBR 322, I told it is TET-R or the tetracycline resistant gene or AMP-R, ampicillin resistant gene. Any one of the gene you can select. So there you will be inserting your gene of interest. Okay, so that's the third requirement for a good vector. Fine. So another method. So you wanted to insert. So either you can insert in antibiotic resistant gene. Okay. So what will happen? You can select whichever the plasmid or whichever the organism carry your gene or whichever does not. The transformants and non-transformant you can able to recognize when you are inserting into the antibiotic resistant gene. Understand? Another method how you will be inserting. Another method is insertional inactivation. What is this insertional inactivation? So have you ever heard about the LAC-Z gene? Remembering the lac-operon? Lac-operon. Okay, so there you have to recollect the LAC-Z gene is responsible to synthesize the enzyme beta galactosidase. Right? So here you can see here. Okay, so wherever the LAC-Z gene is there, so into this particular portion you can keep your insert. Like you can keep your foreign DNA. So what will happen? The LAC-Z gene will not work. It will be deactivated. Understand? It will not transfer. Normally the lac acid gene will be synthesizing the beta-galactosidase enzyme. Okay, so that will be producing blue colored colonies. That's a normal condition. So in this normal condition, what you are going to do, the lac acid gene you are disturbing, or else you are inserting and inactivating that particular gene activity. Fine. So what will happen? It will not be ready to synthesize the beta-galactosidase enzyme properly. Right? So instead of that, what will happen? The translation of your interest, whatever you have inserted will be happening. So that blue colored colonies you will not be getting. Instead of that, what you will be getting? White colored colonies. So here you can able to recognize the white color colonies are the transformants, or else what is that? Your gene or else your foreign DNA has been absorbed by this particular white colored colonies. This is another method of insertion of foreign DNA into vector. Fine. So till now we were discussing the features of a vector, okay, and which are the various kinds of vectors. So we told that plasmids can able to carry this much, cosmids, bacteriophages, bacterial artificial chromosome can able to carry this much, yeast artificial carry how much over the kbs, right? So once it's coming into higher organisms like plants, we have to choose some other guest. Understand? Since the complexity of the cell is increasing, according to that we have to change our choice. Fine. So that's the reason the topic vectors for cloning genes in plants. Understand? If you wanted to clone a particular gene in plants, what kind of vector you will be using? So the exclusive one is TI plasmid. Understand? You know who is carrying TI plasmid? Agrobacterium tumefaciens, which is a soil bacteria. Understand? So this soil bacteria can able to carry this TI plasmid, and they can able to deliver, or else they can able to do the shuttle process. They will be delivering that piece of DNA you have inserted into a plant cell, because plant cell which is having a cell wall, right? That's a specialty of plant cell. Other than any other organism cell, plant cell is protected by membrane as well as a cell wall. So Agrobacterium tumefaciens, which is a soil bacteria which is containing the TI plasmid, is helping us to deliver the foreign DNA into the plant cell. Understand? So they will be inducing the DNA into the plant cell. Tumor induction, that is a TI. They will be infecting the cell. Understand? And they will be producing a tumor-like structure. Through this, the foreign DNA will be going inside. Fine. So Agrobacterium tumefaciens helping to deliver the genes into plants by the help of TI plasmid. Fine. What about the case of animals? So higher organisms I told. So for plants we are taking the help of Agrobacterium. So what's the condition of animals? Plants is having cell wall, animals does not have. So here the retrovirus is the organism helping to clone the genes in animals. What's the meaning of retrovirus? Retro, reverse. Understand? So what we'll do, so they will be, so whichever the DNA of interest or whichever the gene of interest, you can able to see here the wild type retrovirus. So this is the normal genome of a retrovirus or a viral gene. So this normal gene, can you able to see GAG, POL, N. Okay, so this is the normal gene sequence of a virus, retrovirus. Okay, so this normal gene sequence we will be putting our foreign DNA, that is expression cassette, we can say. Understand? So what they will be doing at the time of translation, they will be producing the viral protein plus our gene of interest. So whatever coded here, it will be translating along with the viral protein. Understand? So their RNA, actually the retrovirus is containing RNA is the genetic material. So what they will be doing, once which is infecting the host cell or the animal cell, so it will be converting into a DNA by the help of reverse transcriptase. So that is the meaning of retro. From RNA to DNA, they will be converting their genetic material, and what will happen once it's converting into DNA, it will be part of the host genome. Can you able to see here? It will be part of the host genome. Understand? So this is how the retrovirus helping to deliver the cloning genes into animal cells. Okay, in case of plants it's agrobacterium, in case of animals it is retrovirus. Fine. Now the cloning is done. Now you have to do the transformation. So whichever the foreign DNA you wanted to multiplicate, you have inserted into the vector, either plasmid or cosmid or phagmid, whatever it is, TA plasmid. So you have stitched your foreign DNA. Now you have to transfer into the host cell for the process of multiplication. So that we can say as transformation method. So there are various methods according to the cells. The methods also differing according to the cells, means from lower organisms to higher organisms again. The first method is chemical method. What is a chemical method? So here, if you are taking the bacterial cell, we will be treating the bacterial cells with divalent cations such as calcium chloride or magnesium chloride. Understand? So you will be incubating the cells with divalent cations like calcium chloride or magnesium chloride. So what will happen? It will be making porous. Understand? After that, what will do? We will be giving the heat shock 42 degree C. So that will be making porous like structure on the membrane of the bacteria. So once it's porous, what will happen? The plasmids or the vector will be going inside it. So after that, again you will be recovering. Recovering is through the chilling process. You will be keeping in the freezer. So once it's chilling, again the membrane will be repairing. Fine. So here you can see the cells has absorbed the plasmid. This is a chemical transformation using the divalent cations like calcium chloride or magnesium chloride. Also you can use for chemical transformation, magnesium chloride or calcium chloride. Fine. The next method is electroporation. What is electroporation? So from this name itself you can able to define. Electro means electric shock. Same here, the electrocompetent cells, like they should withstand the electric shock. So the electrocompetent cells will be taking along with that whichever the vector you wanted to insert, that also you will be taking together, and you will be applying slight electric shock. Slight electric shock means approximately 15 kV. Fine. For 5 microseconds, that's the pulse. Okay, so if you are applying this light electric shock, what will happen? The same process like here only, the porosity will be increasing. The porosity of the bacterial cells will be increasing. Once the porosity increasing, what will happen? The plasmids or the vector will be going inside. So after that you will be keeping it for chilling. Then the recovery happens. This is the method of electroporation. What's the next one? The next one we are going to discuss regarding the higher organisms like plant cells. So if it is a bacterial cells, you can use either electroporation or the chemical method. So here the plant cells, if you wanted to transfer into plant cells, what you will do? The most important one, the one is agrobacterium method. That is TA plasmid will be carrying your DNA or gene of interest, and you will be infecting that particular plant tissue. So thus the vector will be going inside, the cloning vector will be going inside the cell. That's one of the method. Other than that, gene gun or biolistic method. So what is a gene gun or biolistic method? So gene gun, which is a gun-like structure only. So here the metal particles, instead of the normal bullet, the metal particles made up of gold or tungsten will be coated with the DNA you wanted to transfer. Direct transfer method. So instead of putting into a vector, what you will do, directly you wanted to transfer a piece of DNA, that you will be coating to gold or tungsten methods, bullets. Okay, gold or tungsten bullets. So what will happen? So once it is shooting, so there's a bombardment of plant tissue with DNA coated particles, they are saying, like you will be coating the DNA into the bullets and will be shooting directly to the plant cells. That is the biolistic method or the gene gun method. The next one, disarmed pathogen vectors. Disarmed means which is not harmful. Understand? Disarmed pathogen like TI plasmid will be using for infecting the plant cell. Understand? So these are all the two different methods, transformation method for plant cells. Of course, for animal cell, what type of transformation method you can use? Okay, so for animal cells, the micro injection. Micro means very small. So from the name itself, we can able to see micro injection, which is directly introducing DNA. This is a method to introduce directly into the cytoplasms. We can able to see here the micro injection needles. Through this needle, you will be selecting whichever the cell you wanted to transfer your DNA to. That cell nucleus you will be choosing. There you will be transferring your DNA to be getting transferred. Understand? That is a micro injection method. So this is only for animal cells, because I already told the plant cells is having plant cell wall. Fine. The next one. So these are all the various transformation methods in genetic engineering. Fine. So now we are going to discuss the important multiple choice questions based upon this particular topic. Fine, we'll see. The question number 1. The TI plasmid is often used for making transgenic plants. Right? TI plasmid. This plasmid found in which organism? So that's the question. The TI plasmid, we know that the TI plasmid is the vector to be used in plant cell. Right? So the option A, yeast. The TI plasmid is a plasmid, they are saying, found in yeast as 2 micrometer plasmid. No. Okay, actually this is an yeast which is having a 2 micrometer plasmid, but we cannot use in transgenic plants. Fine. Acetobacter, you know that, which is a nitrogen fixing bacteria. Rhizobium also a bacteria which is found in the leguminous plants for fixing the nitrogen. So what is that? Which is soil bacterium, Agrobacterium tumefaciens, right? Agrobacterium tumefaciens is the right answer. The TI plasmid is often used for making transgenic plants, so which is found in the soil bacteria, that is Agrobacterium tumefaciens. Number 2. Which one of the following is commonly used in transfer of foreign DNA into crop plants? Crop plants. Understand? So they have given Penicillium expansum, Trichoderma hertzianum, Melodigna incognitia, and Agrobacterium tumefaciens. So what is Penicillium expansum, which is a fungus, right? Penicillium expansum, which is a fungus. Understand? So which does not carry any plasmid. Trichoderma hertzianum, so which is a Useful fungus. So sometimes we are using this as a fungicide, okay, which is found in soil, okay, which is commonly used as a fungicide. Melotina incognita, which is a harmful nematode. So which is a pathogen, okay, that will harmful for the crop plants. So what's the correct answer here? The correct answer is Agrobacterium tumefaciens. Fine. Number 3. Which one of the following is used as vector for cloning genes into higher organisms? Understand? Higher organisms. So if you are seeing here the option A, Rhizopus nigricans. So what is Rhizopus nigricans? Which is a bread mold, right? Bread mold. It's fungus. Fine. So retrovirus will be inserting their genetic material and using the enzyme reverse transcriptase will be making the DNA. That DNA will be part of the host DNA. So this is the right answer. You know that in higher organism, if you wanted to transfer the piece of DNA, you can use the retrovirus as the vector. So what about the baculovirus? Baculovirus is also an useful virus only, but we cannot use in genetic engineering experiment because of their genetic material is double-stranded DNA itself. Understand? So their genetic material is double-stranded DNA. Understand? So there the DNA directly cannot enter into the cell. Fine. Salmonella typhimurium, so you already know which is a what causing organism, which is a bacteria causing typhoid, right? So here the right answer is option B. Fine. Number 4. What is true of plasmid? What is the statement? So out of the statements, which is true for a plasmid? So here they are given the option A, which is found in viruses. No, which is not found in viruses, which is an extra chromosomal double-stranded DNA material mostly found in the bacteria, right? Contains gene for vital activities. Not at all. There is no more vital activities, right? Then which is part of nuclear chromosome? Never. Which is self-replicating, right? Self-replicating independent genetic material. It's not part of the nuclear chromosome of any other organism. And the 4th option is which is used widely in gene transfer as the plasmid we are using widely to transfer the gene. The plasmid we are using as the vector, right? So what's the right answer? The plasmid is widely used in gene transfer. In genetic engineering, the antibiotics are used for what purpose? Understand? In genetic engineering, the antibiotics are used for what purpose? So option A, a sequence from where replication starts. No. Where replication starts? From the gene ori. It's not from the antibiotic gene. To keep the cultures free of infection. Actually this is correct, but it's not the first preference. It's not the first preference. And the 3rd option, using a selectable marker, of course. That is one of the requirement for a cloning vector. Selectable marker should be there. So there only we are inserting. So if this is the antibiotic resistant gene, here only we are inserting our foreign DNA. So thus they are functioning as the selectable marker. To select healthy vectors, no, there is no role of healthy vectors. Nothing. Fine. Next one. For a DNA to function as a cloning vector, the most essential requirement Understand? So think that you have a double-stranded DNA. Okay, you have a double-stranded DNA. You wanted to use this as a cloning vector. So what is the essential requirement you have? Multiple restriction site. So this we already told. No, multiple restriction site, what will happen? It will be leading to fragmentation. Your vector will be breaking into so many pieces. That should not happen, right? So that's not the one. Then several selectable markers, that also not possible. It does not want several selectable markers also making issue. Understand? So either one or two. Few, very few we needed. Few selectable markers we needed for genetic engineering. Circular nature. Circular nature, it's not necessary. Right? And the fourth one, that's the important one, right? Ori sequence, or the origin of replication. So definitely the vector you are using should have origin of replication. Then only the number of copies you will be getting. So what is the answer here? The option D, Ori sequence. Fine. Number 7. Bax and Yax are. So what is Bax and Yax? Bacterial artificial chromosome as well as yeast artificial chromosome. So we already told they can able to carry like 1000 kb and 300 kb of the foreign DNA. Okay, so regarding Bax and Yax, which statement is correctly matching? Number 1, natural DNA obtained from bacteria and yeast. In their name itself they told bacterial artificial chromosome and yeast artificial chromosome. So here the natural word itself does not require, which is an artificially made one. Next one, which is useful vectors for eukaryotic gene transfer. Correct, because they can able to carry a large number like this. Eukaryotic gene transfer they can able to do. Okay, we'll see the next statement also. Artificial DNA obtained from bacteria and yeast. Of course, they are the piece of artificial DNA obtained from bacteria as well as the yeast. Correct. And if you are seeing the option D is saying both the statements B and C are correct, of course. Right? B and C are correct option. Next one. Transgenic plants are produced by. What is the meaning of transgenic plants? Trans and genic. Some genes you are transferring. From this name itself we can able to enumerate. Right? So you will be getting the transgenic plants. Transgenic plants are getting through inducing gene mutation, not anymore. If you are mutating the gene, we cannot guarantee your plant has been changed. It will be changing the characteristic features because of mutation, but whichever the desired quality we cannot say. Right? So inducing gene mutation will not give transgenic plants. That's wrong. Arresting spindle fiber formation. So at the time of cell division, the spindle fibers, okay, the spindle fibers is getting arrested. No. Transgenic plants, if you are doing something at the time of cell division, you will not get any transgenic plants. That's wrong. Deleting sex chromosome. So if you are deleting the sex chromosome, what will happen? The characteristic feature of the plant itself going to change. Right? The fourth one, introducing foreign genes. Of course, correct. So the transgenic plants are produced through the introduction of foreign genes. Fine. Biologistics or gene gun is suitable for what? Biologistic or gene gun. So you already know biologistic or gene gun, which is a transformation method. Okay, so we'll see the options. Constructing recombinant DNA by joining with vectors. So here, constructing the recombinant DNA by joining the vectors. Who is doing that? That's a function of an enzyme only. The ligase will do that function, right? You do not want to use the gene gun here. The ligase enzyme will be joining the DNA with the vector. Disarming pathogen vectors. Disarming pathogen vectors, that is the TI plasmid. So will be directly infecting. Do not want to use the gene gun. Next one, transformation of plant cells. Of course, this is the correct option. So you wanted to do the transformation. So directly you will be taking the DNA, right? DNA you will be coating into the bullets, right? The bullets made up of gold or tungsten, right? Gold or tungsten, and directly you are bombarding into the plant cells. So biologistic or gene gun method is suitable for transformation of plant cells. So DNA fingerprinting does not related to gene gun, right? Number 10. Electroporation procedure involves what? Electroporation. So electroporation is also one of the transformation methods. So in that method, what else has been involved? First one, fast passage of food through sieve pores in phloem elements with the help of electric stimulation. So they are saying the passage of food through sieve pores in phloem elements. So here there is no need of electrical stimulation, right? So it's a natural process, the passage of food through the sieve pores and the phloem elements. All these things are happening naturally. We wanted to use the electric stimulation. That's a wrong statement. Opening of stomatal pore during night by artificial light. That also not needed. Artificial light opening is happening, but it's not through the process of electroporation. Understand? So here they will be using some light, okay, that is photolight. Particular bulbs are there, so they will be using that particular temperature bulb. So under that the plants can able to open their stomatal pore under laboratory condition. So however they are studying the photosynthesis process using by this particular method. Making transient pores in the cell membrane to introduce gene construct. Of course, that's a correct statement. So what will happen? So in electroporation method, I already told you 15, what is that? 15 kV, right? 15 kV. Small light electric shock will be making what? Porous, small porous, transient pores on the membrane. So through this membrane what will happen? The gene whichever you have constructed, you wanted to introduce will go inside. The next one, purification of saline water with the help of a membrane system. No, never. That's a osmomembrane system, membranic reaction system. In water filter, whatever you are using, it's not electroporation. Fine. Next one. Which of the following is the example of direct gene transfer? That is, only the particular gene you are transferring directly. So, like you know that the cloning vector. To the cloning vector, you are introducing your gene and transferring. Other than that, some of the methods are there. So this is the piece of DNA. Okay, this DNA you can directly introduced into the host cell. So they are asking which are all the direct gene transfer methods. So you know micro-injection. So here what's happening? You are injecting the gene of interest into the nucleus of the cell, right? Nucleus of the cell you are directly. So that's the correct answer. So if you are seeing the electroporation, what is electroporation? So here, so in a tray you are taking the cells whichever you wanted to introduce, okay, and the piece of DNA you want to insert. Here what you will give? A light electric shock. Light electric shock. So through this shock what they will be making? Transient pores, right? Transient pores they will make. So through these pores the DNA will be directly going inside the cell. So which is the direct gene transfer method. Particle gun. So here also the same thing. What is the particle gun? So particle gun is a method of coating the gene or the DNA of interest, right, into the bullets and bombarding into the cell. That only the particle gun. So here we can choose the option D because it includes everything, all the above. Fine, understand? Next one. Cosmids can clone DNA fragments up to, that is the, what is the carrying capacity of cosmids? That's the question. So they have given 45 kb, 50 kb, 85 kb, 75 kb. So first of all you have to think cosmid is equal to phage plus plasmid. It's a hybrid vector. Phage plus plasmid, that is 25 kb. If the phage is taking, 20 kb is the capacity of plasmid. So how much a cosmid can carry? 45 kb, right? So what's the answer here? Option A, right? Bacterial cells are made competent by treating with what? Okay, so bacterial cells, okay, so this is a bacterial cell. So you wanted to make the transient pores on this, okay. So here you have your vector. So you wanted to introduce into this bacterial cells. How you will be making competent? How you will be getting absorbed? Okay. So in transformation method we told that is the chemical method, right? Chemical method of transformation. What is that? You will be treating your bacterial cells with divalent cations like calcium chloride or magnesium chloride. Any one of this you can use to make competent bacterial cells, right? So what is the answer here? Divalent cation of calcium. It's a chemical method of transformation. Fine. Next one. Insertional inactivation is the process of silencing which gene? Can you able to see? LAC-Z. LAC-Z gene is responsible to produce which enzyme? Beta galactosidase, right? Beta galactosidase. What about the next one? Origin. Responsible for replication. Origin of replication. What is ROP gene? What is ROP gene? Replication of protein, right? Replication of protein. TET-R. It is an antibiotic resistant gene. So here if you are inserting and silencing which gene will be getting a transformant colony. So it is a checking point using as a selectable marker. So you already know beta galactosidase is getting secreted by lac-Z. So to this lac-Z, if you are inserting this lac-Z, we can be getting silenced, and thus the production of beta galactosidase is getting interfered, and you will be getting the white colored colonies. Thus you are identifying the transformants as well as the non-transformants. Understand? So here what is the correct answer? lac-Z. Right? Insertional inactivation to particular lac-Z gene, you are inserting the foreign DNA and inactivating the action of that particular gene to identify the transformant colonies. Fine. So that's all for the day. So these are all the important multiple choice questions from this particular topic. So thank you for listening me. Have a nice day. Bye bye.